Again, thanks for joining us tonight. My name is Shelley. I'm an admissions rep here at Alma College. And tonight we're doing our second kind of workshop with our pre-health professions. The first one, we were able to introduce all of the different faculty that were part of our group and lots of information about the different offerings that we have for our pre-health professions students at Alma College. And then today we're going to actually give you show some tours of some different labs on campus and off campus. And then also I'm going to have some presentations by some students, current students, having them show you some of their research opportunities that they've taken place. So we'll go ahead and get started. This is recorded, just so that I want everybody to know this is going to be recorded again. So we'll be able to have this to follow up with. But we'll go ahead and start sharing my screen, and we'll start with our IPHS Integrated Physiology and Health Sciences tour.
So we are standing in the Human Performance Lab, and this is the main lab space that IPHS, or Integrated Physiology and Health Science, uses for our laboratories that we have in class. We do have a few other labs. We have a cell biology kind of lab where we plate some muscle cells and we do experiments on them. We also have an EKG testing and biomechanics lab, but we're going to focus on this one for this tour. Now this area is kind of a seating area. You can see that we have a chalkboard over on the side, and we have this big screen TV. We use this for journal clubs that we have on Fridays during the months, where students can come and talk about current research or present the research that they're doing. At these tables, we also do the majority of our physiology labs. So we normally will have 4 setups where we have a computer and BioPack systems that allow us to measure different physiological variables, such as lung volumes by breathing into a spirometer, or looking at heart rate, or looking at how quickly we can react to a stimulus, so neuroconduction velocity or reaction times. Now this is a pretty decently sized lab, and if we come back in the back portion of the lab, we have a lot of exercise equipment, which you will see. Some treadmills, some bikes. That's because this is the equipment we will use in exercise physiology. So after we learn how the human body normally works, we start talking about how does it work when you add in stressors, such as exercise. And so students will get on the treadmills and run, or get on the bikes, and we can actually measure how much oxygen they consume, which can tell us things about how many calories they burn, or their fitness level, because the more oxygen you can consume, generally the more fit you are. We can look at lactic acid production and see what is the time at when they produce lactic acid. So what is their threshold, which all tells us information about how they handle that stress, or how fit they are. Now as we come back here, you'll see this kind of large chair region. We also use this for exercise physiology, but we use it for biomechanics when we start getting into the physics of human movement. This allows us to look at velocities with which we can move our leg or our arm. It also allows us to measure forces that we can produce from the muscles in our leg, our arm, our torso, because we can set this up to allow us to push with our lower leg, to push by extending our leg. We can set it up to look at wrist movement. We can set it up so this chair is actually laying down and someone could do movement where they're lifting their torso and looking at force production in their abdomen. Now as we move back here, we have a little room off to the side, and I will walk in here. This room has this tub-like structure in it. This is what we call the hydrostatic weighing tank. We can fill this basin up with water and have someone completely submerge themselves in the water, and we can determine their body composition. So what percent fat, what percent lean mass, such as muscle, how much water content do they have in their body. And then if we go into this final laboratory space, which is part of our human performance lab, this is the cardiovascular section of the lab. We don't have all the equipment in here right now, but you'll notice this hospital bed right here. This is in here because Dr. Branish in our department is an expert in echocardiography, and in particular looking at blood flow dynamics. So she can set someone up in this bed, get her echo machine in here, and start looking at blood flow through the vessels. And then this last piece of equipment in the cardiovascular lab is what we call the lower body negative pressure box. We can have someone actually get in here to where their torso from kind of mid-torso down is in the box. And then we can put them in a nice kayak skirt. So I can put this around my waist. We hook it up to this box so their upper body is sticking out. And then we actually use a vacuum to create negative pressure in the box, which pulls the blood down towards your lower limbs. And we can look at how your heart responds to try to maintain blood pressure at a normal number. And that's kind of the main equipment that we have in our human performance lab. But you'll also see a lot of students in here doing student theses, independent studies that deal with a lot of different questions that could be asked about human physiology. Now, when we look at electromyography, we're actually looking at the electrical activity in a muscle because muscles are excitable tissue. Now, to get that electrical activity, we have to use electrodes. So we're going to use the BioPack, and you can see that we have set up basically 2 sets of electrodes. Each set of electrodes will measure the electrical activity or the EMG activity from one muscle. We're going to do the quadriceps and the hamstrings today. And a little bit later on, you're going to see the setup on the leg, how we actually put those electrodes, which you see right here, on the leg, and then clip to them to actually measure. Now, we are using the BioPack system. We have a modified version of the EMG lab. It's called Double EMG over here, and it's saved on the desktop. I have turned the BioPack system on. You'll remember this is the BioPack, and the on switch is on the back. And you will know it is on when you see a green light by the power button right there. So once the BioPack is on, we open up the program on the computer. We start this double EMG lab over here. And when you have the person, the subject, set up with the electrodes, all you have to do is press start. Now, when you first open the program, it's going to look like you only have one channel, one set of electrodes being recorded. However, this is set up so that when you press start, the second channel should jump on the screen. And now you see that. So now we have channel 1 and channel 2. So whatever muscle group you put the first set of electrodes on will show up here. And same thing for the second set of electrodes. Now, what we're going to do is we're going to have people perform activities. In the example later on, we're going to have someone ride on the bike. Dr. Montoya will ride on the bike. And you should see changes in the EMG levels of each muscle. Take a little pause. We wrap some ACE bandages around to hold on to the electrodes as Dr. Montoya cycles. And now we can see this beautiful plot of the EMGs. In the red on the top, we have the quadriceps. In the blue on the bottom, we have the hamstrings. We're increasing the intensity a little bit. And now you can see what happens to the EMG traces. They get larger, which indicates greater electrical activity in those muscle groups or greater activation of muscle fibers.
So we have just calibrated the Parvo metabolic analyzer. We're going to perform a resting metabolic rate measure. So Dr. Harfman has so kindly agreed to be part of this. So you can see we have the mouthpiece set up. She's got that, and then she has a nose clip on, those blue things. That means that all the air coming into and out of her body is coming through her mouth. So we can measure it, nothing is escaping through her nose. So all air that she inspires comes from this side, the ambient air, and then all air she expires is going to go through this tube, which we have not hooked up yet, but we will right now. So you can see that set up. So for resting metabolic rate, in a perfect world setting, you would bring somebody in fasted, meaning they have not exercised or consumed calories for at least 12 hours. And we would then have them lay here supine for about 30 minutes or so while taking the measure. So we're trying to get as close as we can to a true minimal energy needed to sustain life. Okay, so we're going to be starting our testing. So I just hit the OK button once we had all of her information input. And so this will take about 10-15 seconds. And then it's going to say OK to start testing. So we'll sample here. and it will take about 15 to 30 seconds to display the first data. And when that displays, I'll start pointing out some things to focus on. So again, you can see the setup. She's lying down. We've got the tube. Now instead of hooked into the syringe, it's hooked up to the mouthpiece so that all the air she's breathing is being fed into the metabolic analyzer. And then we can start capturing data. Okay, so here's our first reading then, and it'll update every 15 seconds. This is just the time. This RR stands for respiratory rate, how many breaths she's taking per minute. Her VO2, so the amount of oxygen she needs, is right here. This is liters per minute. She's at 0.15, which if you express that in relative terms, so relative to body weight, is 2.6 right now. Her ventilation, or VE, is 4.4 liters per minute, so that's how much air is moving into and out of her body on a minute basis. Her VCO2, so VO2 is the amount of oxygen you consume. VCO2 is the amount of carbon dioxide your body is producing. She's at 0.15 right now. And then this RQ, you see it in two different spots. RQ is calculated by dividing CO2 divided by O2. So 0.16 divided by 0.19 would give you 0.85. RQ, or RER, is a way of determining fuel utilization. This number ranges between 0.7 and 1.0. The closer that you are to 0.7, the greater your utilization of fat. The closer you are to 1.0, the greater your utilization of carbohydrates. So you can see Dr. Harfman is almost exactly 50-50 split between fat and carbohydrates as her energy source right now. This data gets recorded, you can see down here, every 15 seconds or so. And then after about a half hour of doing this, we would print this out. We would take some averages in order to understand exactly how much energy she needs just to sustain baseline functioning.
All right, thank you. Dr. Hartman, you want to talk a little?
Yeah, hopefully that gives you a little bit of insight into the types of labs that you might do in the IPHS department as students, except that as the students, you would be the people using the equipment and you would be the individuals actually performing the activities in those labs. I'm going to turn it over to Haven Westra now. She is a student who has done a lot of those laboratory experiences, and she's done some research in IPHS with Dr. Montoy. So take it away, Haven.
My name's Haven Westra. I'm currently a senior. I did summer research last year with Dr. Montoy, who was the other professor in those videos. And I'm going to be presenting my research that we did. And the title of it is, Can Your Face Affect Your Pace? The Impact of Facial Expression on Running Economy. So we did this study last summer over quarantine, completely remote. So we took some data and methods from some students at Hope College with Dr. Brian Ryder. And I was able to still get the summer research experience safely. So just some background information that I think will be helpful is, what is running economy? And running economy is the amount of energy required to maintain running at a given speed. So if you have a strong running economy, you use less oxygen to complete a task. And this study was a replication study, so there was already the methodology out there, and we repeated it to see if we got the same results or not. So we replicated Brick et al.'s 2018 study, and they found that if you smile while you're running, you can increase running economy by 2.8%. And they thought this would be true because as you're smiling, you relax more and you could decrease muscle activation, resulting in a decreased energy output. So as you can see here, there were a lot of news outlets that took interest in that, and that's kind of what brought our attention to this topic. And the purpose of this study was to replicate and expand on the Brick et al. Study, and we hypothesized that smiling would result in better running economy, like Brick found. As I mentioned, the subjects were Hope College students, and they were women's soccer players. So that was our participant demographic. As you can see in these pictures, we began with fitting the mask. This is a different style of mask than we have in our lab. The one that Dr. Harfman had the nose plug and the mask you put in your mouth, but this is just a different style that does the same thing. And they took like a baseline test on the treadmill to get their 70% VO2 submax. And then they came back again and did 4 6-minute blocks of running. And they did each of our 4 attentional cues. So you can see the subject on the left smiling with the mask on, and then frowning with the mask on. And then our cue for relaxing was kind of like pinching your finger. And then the 4th was just the control. And as you saw in that last video, that metabolic analyzer produces a lot of data. So that's what we used. The primary variables were VO2, heart rate, perceived effort, which they gave like subjectively, and then blood lactate. So we measured all those outcomes to kind of determine if running economy differed when they were doing each of these cues. And we have the chart down in the bottom, time on the x-axis and oxygen consumption on the y. And as you can see, it's kind of misleading because it looks like the smile had a lower oxygen consumption, but when we did our statistical analysis, it wasn't enough to conclude significance. So our study was contradictory to Brick et al.'s and suggesting that facial expressions do not result in improved running economy. And this is important because it leads us to believe that running economy is training induced and not affected by acute manipulation like smiling or frowning or relaxing. And another reason why we thought this study was important, because of the amount of press it got, was that if coaches are telling their runners, hey, it's super important that you run, it will make you run faster, that's not scientifically founded in our experiment. So we wanted to make sure that athletes and people who are training for marathons or whatever are using scientifically supported methods. And in my summer research experience, I used so many of the skills that I learned in my physiology classes and my classes about how to conduct research and how to prepare a paper that would be ready to be submitted and accepted by scientific journals. So again, I'd like to thank Dr. Montoy for selecting me to be his research assistant, and Dr. Ryder and his students for getting us started on this project. Thank you.
Great, thank you so much, Haven. Alrighty, I'm going to go ahead and share my screen again, and we're going to show you our bog here at Alma College. It's just off campus. It's a great research opportunity for students of biology.
Hello, I'm Dr. John Rowe, and I am at the Alma College Ecological Station out in Vesterburg, Michigan. This is an extension of our biology campus about 12 miles west of Alma. It is a 230 some acre plot of land of upland forest, old field, just what you can see behind me, lakes and streams and wetlands. Here we are at the Alma College Ecological Station. Right, we have arrived at the station here. Here is the research building, research and classroom space at Alma College Ecological Station. And here is our classroom space. Of course, there's some various office space for faculty and students to work. See, there's trails here that go around the entire lake. Dogs out here through the day. And if you look right through here, you can see the snowy frozen surface of the lake. Right through those trees, a little bit in the distance there. Here's the snowy pathway down to the lake. come down a walkway. But you can't see it. It's a plastic walkway. Takes us out to a dock here. And here is a very snowy, frozen Davis Lake.
So let's rewind to Davis Lake during summer. And during summer is where a lot of our research opportunities are available for our students. There have been a number of studies on the forest, on a lot of botanical studies, limnological studies, studies in herpetology, which are reptiles. These two particular students are out doing their daily surveys for snakes. And you study the ecology of snakes. This is a northern ribbon snake. We also look at garter snakes and water snakes as well.
56. So 5 meters west of Centrifugation.
Here is a northern ribbon snake. You see her head up to the left there, kind of hidden. She is presumably up regulating her body temperature. It's about to take off. So as you saw, we have a research classroom facility. The ecology course has been taught out here, medicinal plants have been taught out here, a lot of natural history courses have been taught out here. And here, students are back doing their lab research and putting electronic tags in the snakes for future identification. So Alma College has a number of opportunities for research, both on campus and out in the field at the Alma College Ecological Station. The objective of our exercise here will be to assess the relationship between metabolic rate, as measured by oxygen consumption, and body size of various ectotherms, including frogs, lizards, snakes, a tortoise, a slider turtle, and a tarantula. The reason we're doing this is we're going to attempt to discern whether we can detect a relationship where metabolic rate on a per gram basis is highest in small individuals and becomes smaller as individuals get larger. So as individuals get larger, their metabolic rate tends to decline. So we see this relationship in birds and mammals, and the reason this occurs is that the high surface to volume ratios of small individuals promotes energy loss and thermal energy loss. And so smaller individuals must maintain higher metabolic rates in order to maintain body temperatures sufficient for metabolic reactions. We even see this pattern occurring in ectothermic vertebrates such as lizards and amphibians. The trend can be detected across species. So here it is from our smallest mice, very high metabolic rates that tail off into lower metabolic rates in larger individuals.
Okay, so we're going to be measuring oxygen consumption in this small frog. This is a green tree frog from the southeastern United States. Okay, you can see it has toe pads and allows it to cling to things. And we're going to be putting this frog into a respiration chamber here where it will sit. It's a sealed system. We're going to have our oxygen sensor measuring the oxygen consumption of this frog inside this little chamber.
So here, the students are obtaining mass of the snake.
Let's see what it says. So I got 513.6. 513.6. Yes, I did. Okay, good. 513.6. 513.6.
Following the determination of the mass, they determine oxygen consumption of the snake. The students are weighing a tarantula for their work. We also did work on a Greek tortoise. It's one of our larger ectotherms. And finally, the small red rat snake is one of our smaller ectotherms.
All part of the experience, you guys.
Go, stop. Okay.
I'm not trying to do that. I just think they're cool.
10.1.
Thank God.
With subsequent determination of oxygen consumption. So if we look at some of our data here, is mass-specific O2 consumption across our vertebrates, and we can see that on a per gram basis, pregates are maintaining relatively high metabolic rates, and it trails off significantly so when we get out here to the larger vertebrate ectotherms. If we look at just the vertebrate ectotherms, we can see that that the smallest reptiles and frog, the small anolis lizard, had very high metabolic rates, and it trailed off into the smaller snakes, Transphyla, and out to the medium snake and large snake, and finally out here to the tortoise. So we were able to detect that negative relationship between mass-specific oxygen consumption as a measure of metabolic rate and body size of ectotherms.
All right, thank you, Dr. Rowe. You want to do a kind of quick little recap there?
As you saw, we have the BOG facility, and I showed you some of the things that we do on campus and in the physiology class. But we also have full lab facilities on campus. We have a microbiology and cell biology lab and places for molecular work as well. And we have research opportunities in those areas as well. So we've got a number of research opportunities and teaching and learning opportunities in molecular biology, cell biology, and into organismal biology and sorts of things that I showed you. So with that, I think I will transition into Kaylee Prewitz, who is going to tell us a little bit about some of her laboratory-based research.
Yes, thank you, Dr. Rowe. I love watching that video, but the spider gives me nightmares like every time I see it. So while you're doing your organismal research, I'm more focused on the molecular genetic side of it. So let me see if I can share my screen. Okay. So my name's Kaylee Prewitz. I'm a senior here on campus. I'm a biology major and a chemistry minor. And what I did with Dr. Calhoun in his research lab the summer of between my sophomore year and junior year was work on human variation. So what I was doing was trying to figure out what single base pair change in the genetic combination would yield a different phenotypic or visible characteristic change. So some examples that we first started out with researching, which was the #1 starting point, I think. So some examples that we, I guess, looked at would be cilantro. So if you were to have a letter in your genetic code and it being A, you might think cilantro tastes like soap. But if you were to have the genetic letter being T or something, you would taste it like it would be the herb that you think it is. So some other examples would be if you can taste PTC paper, as well as what your blood type is, as well as your eye color. So these are just some important characteristics that contribute to the diversity between you and me. So I guess I want to take you through, I guess, the step-by-step process of what I go through in the lab today. So let's see. So I guess the first step would be research, but then once we figure out what gene we want to go for, we need to make a primer for it. So what we do is we figure out what 2-letter combinations you could have. So if I was an A and you were a T or something, I would need to make a primer that would bind to it. So we do this by synthesizing something that looks the exact same as our code. And right here, I'm showing my O- blood type. So if you were to have an O- blood type, your genetic code would look like this. However, if you were a non-O blood type, your genetic code would look like the bottom. So in order to make primers that would bind to each of these two things, we need to come up with a complement. So that's exactly what our primer is. So moving on from that, our next step is to isolate our DNA. So I think my favorite part about working in the lab was that you get to physically see more about yourself in this aspect. So when I was testing myself on all of these different genes, I was using my own DNA. So how we did that in the lab was we did our swish and spit test. So we would take a vial of saline solution. So it just tastes like salt water really. And we'd swish it for maybe 30 seconds and expel it back into the tube. And then we would boil it in order to split the cells apart and then centrifuge it, which this is what this machine is showing here, this picture. And then we would get our precipitate being your DNA cells. So on our primers that we make, we're able to use our DNA to match it. So then we move on to the next step, which is PCR amplification. So while we mix all of these ingredients into the tubes with our DNA, so with our DNA, we add our primers, we add all these other ions to help it bind, the primer bind to the complementary DNA. But once we get there, we need to make sure that our binding is, I guess, accurate. So how we would do this is PCR amplification. And that's what this machine over here, our mini 16 machine, and this is like the top-notch technology, guys. Super smart. It's hooked up to our phone via Wi-Fi, so I can walk around with it, and it even shows like animation at the bottom of it. So what we do is our first step is denaturation. So with our DNA, we need to split our two DNA strands. And then our next step is annealing. So we need to anneal our primer to our DNA strand, and then we need to extend. So we need to keep amplifying our products. So you can see here that we start with one copy, but in the end, after just what, 4 replications, you're getting 16 copies. This is going to do this hundreds and hundreds and hundreds of times by changing the temperature of our samples, I guess, in order to get a, I guess, trillions and billions of product, basically. So once we have our product, we're able to, I guess, visually see it by gel electrophoresis. So what we would do here is what my picture is showing on the side. We would make a gel, which is just what you're thinking of it, basically, a gel square with little spots for our samples. And our gel electrophoresis machine is going to pump an electric current through our gel in order to push the DNA to one side or the other. And our DNA is negatively charged and opposites attract, so it's going to attract to the plus side of the gel, making our samples run along the gel. So you can see our product here on the left. This is something that at the very end, this is like what you want. So you can see my genes, and these are for eye color. So all 6 of these genes, or all 5 of these genes, contribute to your eye color. And each one is going to have a probability. So I know that my HERC2 gene is mainly what tells me if you have brown eyes, versus my SLC45A2 gene is going to contribute more to blue eyes. So I have my 2 primers here, and it looks like I have my allele for T. So if you were to have a T allele for your HCRTC2 gene, you'd be more likely to have brown eyes, versus if you had the C, which you can see I don't, my DNA did not bind to my C primer, so I have blue eyes. So it's kind of like a check in a way, which I like because you kind of already have the answer. And you're just kind of trying to prove that the gene matches up with your specific trait that you're seeing. So some ways that this is important, I guess, is number 1, forensic analysis. So if we could isolate some DNA found in a crime scene, we could essentially tell you what the perpetrator would look like. So we could tell if he had brown eyes or blue eyes, or if he had a certain hair type, what color his hair is, if it's curly, if it's dark, or whoever, basically. And I guess another one that we use PCR purpose for is COVID-19 testing, which is also important. So that's another good one. And then other human variation analysis. So like our cilantro, our PTC paper, and blood typing. So very cool. I will hand this back over to you guys.
All right, thank you so much, Kaylee. We are going to go ahead and show some of the lab in our chemistry area.
Oh, hey, sorry. Hi guys, my name is Dr. Turk and I am the chair of the Department of Chemistry. And over here we do all kinds of cool chemical reactions and blow things up, and well, maybe that'll come later. I'm an organic chemist. We have organic chemists, inorganic chemists, physical, analytical, physical, and we all do unique things. And since my office is kind of boring, why don't we go walk around and see what some students are doing? This is one of our organic chemistry labs. These are students in their first year, freshman, second semester. Come on down. So what they're doing is they're purifying and isolating a compound within a mixture. So they're weighing some things out here on this side. Over here, they're doing a technique called column chromatography, which requires the little white powder in these long glass tubes. They're just getting started. If we come around over this side, you can see we've got a nice TV screen that our instructor is using to display the experiment for the day and get some tips. Now let's go look at some cool instrumentation that they get to use. This is one of our instrument labs, and when the students perform their chemical experiments, they oftentimes come inside and they determine what the purity is. They do some quantitative work. So we have an infrared spectrometer. You know, back in the day, these things were this big, but now look at the thing. It's like cell phones, they're tiny. That's that. Here we have a series of GCs, or gas chromatography instruments, that are used to separate gaseous mixtures. And this is really the same thing here, but it's a bit more expensive, probably about the cost of my car. This one will actually do some data analysis and determine the identity of a complex mixture, something that you might see on the show CSI. This device here is called the Spectramax I3x, which probably doesn't mean anything to you, but it's a plate reader that measures the fluorescence of molecules. And so the way that I use this in my research is I measure the inhibition of influenza virus. It's human influenza virus. We make little tiny molecules with fluorescent tags on them, and as they interact with the virus, we see changes in fluorescence, and this thing can measure it. It's awesome. Let me show you where we keep our virus. This is our -80 freezer. It is really cold inside here. Whoa. And it's down in here, way in the back over here. You see that N1 right there? It stands for H1N1. That's the neuraminidase virus. You probably heard about that on the news, but we use that right here in our research lab. Let's close that back up. Hey guys, this is where we do our viral testing inside of here. We'll turn the lights on. Come on inside. It's not that dangerous. We've got a laminar flow hood right here that sucks the air out of the room while we're working inside. It's very, very safe. And inside of here, we keep some of our ultra cold materials as well. Other than that, this is a really cool place to be. This is probably the coolest instrument if you're an organic chemist or any synthetic chemist at all, like I am. This right here is about a $500,000 magnet. And it can actually interact with the nucleus of different atoms, causing them to spin, and the computer measures an absorption, and bang, we can determine the structure of the molecule. Come on a little closer. So it's a superconducting magnet. It actually has liquid helium and liquid nitrogen around it, this big giant dewar. The magnet on the inside is only about this big. And so right inside here is where we put our chemical samples. If you were to take this instrument and make it really large and lie it on its side and climb inside that hole, that would be an MRI. So NMR stands for nuclear magnetic resonance spectroscopy. The MRI at the hospital stands for magnetic resonance imaging spectroscopy. Same thing. About every 2 or 3 weeks, we order 265 liters of liquid nitrogen to feed that beast and keep it cold. That's why they call it a superconducting magnet, because we keep it so cold. One of the cool uses for liquid nitrogen is right over here. Our biology department and biochemistry department use this right inside here where they keep these cell cultures ultra cold. So here's one of our biochemistry labs. Our students generally take biochemistry in their 3rd year, sometimes in their 2nd year. It's kind of messy in here, but boy, these people do some really fantastic work. They all are tending to work independently on projects. Almost all these stations are set up the same with fluorimeters, their own centrifuges and vortices. all kinds of cool stuff. This is a big ultra centrifuge, which is used to spin things round and round and round and round and extract out proteins and cells and all kinds of neat stuff. This is another one of these really cool instruments we have in our biochemistry lab for infrared imaging. I have no idea how to use it. This is an INS5 semiconductor sequencer. I have no idea what that is. And that's a microwave. I know how to use it. So this is a great place, especially when it's filled up with students, but why don't we go try to find a place where there is students right now? Okay, this is our Chem 115 laboratory, so it's filled with freshmen, very first semester course. Well, I guess it's winter semester, so these are second semester freshmen, so come on inside. A lot of these students just came out of first year seminar, and this is their first science experience. They're almost all done with their chemistry, but we found someone over here. They're actually measuring the density of soda pop. How cool is that? Diet Coke, root beer, not Sprite, right? All of our undergraduate laboratories are staffed by faculty. We don't have teaching assistants running the labs. This is Dr. Mazuka, our physical chemist. I have no idea what he's doing over there, but you know, it's got something to do with the density of soda pop, which is really cool. I found another group of students over here. This is the same class, instrumental analysis. We saw earlier Dr. Straight was leading the class. Like I said, there's always a faculty instructor in your lab. But here we found a TA. She's a chemistry major, Nikki. And over here, they got some really cool solutions that they're making. You know, all kinds of nice colors here. They're generating some, I don't know what. What are you guys doing over here?
These students are generating standard solutions of copper to run in the UV vis to form a solution, to form a calibration curve for their soil samples.
If I wasn't a chemistry nerd myself, I'd just say you nerded out. But that was great. Good job. Okay, so this laboratory houses some of our chemistry seniors and high-level science majors. They're working on chemical instrumentation, and I actually know what they're doing, so come on inside. This is Raquel and George. They're working on a high-performance liquid chromatography instrument. It was donated here just recently. This is a state-of-the-art instrument. What are you guys working on?
So we are looking at finding different metals in our samples, soil samples, particularly looking for copper samples.
What she said. So here's another group of students that I think they're working on the same soil samples. Christina, tell us what this is.
This is electrochemistry. So we put an electrode inside a sample that we took out all the oxygen in because the oxygen will react with the metals.
So is this something you do to monitor river water or soil? Is it just soil samples?
Yeah, it's a soil sample that we diluted.
How cool, thanks. Okay, so this group of students is talking to Dr. Straight, and we don't want to get in their way, but come a little closer. So this instrument here can actually measure metals, again, in soil and samples. There's a big furnace over here that ignites a sample and it lets off a color based on the metal. Like, you ever seen fireworks glow red and orange and purple? It's all based on metals, and this can measure it. Oh, you're back. Sorry. Hey, thanks for coming out and watching my tour. We got a lot of great things going on here, and I hope to see you next fall.
Most exergonic reactions release heat energy, or enthalpy. Chemiluminescence reactions, however, release energy in the form of light. Luminescence created by biological chemical reactions are very common. This molecule of luminol also provides a luminescence when it reacts in the presence of hydrogen peroxide, base, and a catalyst. The result is formation of a high-energy intermediate, which, when that intermediate relaxes back down to its ground state, releases energy, here again in the form of light. We'll want to bend a Pcr pipette at about 90 degrees so we can use that inside of our reaction test tube to aid in the removal of water. Also, we should begin heating water, as we're going to use that at the end of our first step. To our reaction test tube, we want to add 200 milligrams of 3-nitrophthalic acid, 400 microliters of 8% aqueous hydrazine, and our solvent for the reaction is here 600 microliters of triethylene glycol. Maneuvering the test tube, Pcr pipette, and clamps all synchronously so that we don't break the pipette tip is a little complicated, but once it's in place, about an inch or two above the liquid, we want to begin heating gently so that our boil isn't so aggressive that we are removing it by the suction of the pipette. Remember, it's only there to remove vaporized water, which you can see here being generated in the test tube on the sides. After a few minutes of consistent boiling, our original pale yellow color will begin to turn a little bit darker yellow, a little bit brighter. As we hold a steady boil for a few minutes, the color will transition from pale yellow to a golden amber color, sometimes taking on a slightly orange or pink color. Once the reaction is cooled, we now add hot water to the reaction, 3 milliliters, and what we're doing is reducing the solubility of the product and therefore precipitating it from solution. After mixing and cooling further in a beaker of water, we're ready to proceed to centrifugation. Centrifuging the reaction test tube at high speed for no more than 30 seconds will press the solid, heavier material to the bottom of the test tube, separating it from the liquid and allowing us to decant. Decanting is the careful separation of liquid from solid. The residue that remains in the test tube is ready to move on to the next step. That is our crude 3-nitrothiol hydrazide. To this we add our 10% sodium hydroxide, vortex until dissolved, and then add 600 milligrams of sodium hydrosulfite. Next we go back to heating in the test tube with our Bunsen burner, and this time we're just heating to just below boiling for about 5 minutes. This will bring our reaction to completion, whereby we can then add 400 microliters of acetic acid, cool the tube in a beaker of cold water, and then again collect the precipitate by centrifugation and decanting as we saw before. With the solid remaining in the test tube, we're ready to move on to create our individual solutions, and then in a darkened room, pour them together and look for the luminescence. Here's the resulting luminescence from 2 students. Sometimes the color varies a little bit, but more often it's the brightness, and that variability has most to do with the relative purity of the samples.
All right, Dr. Turk, you want to take us, kind of recap that a little bit for us?
Sure. Hi, everybody. Thanks for coming tonight. I was right in the middle of a class and I double booked myself, so I apologize. But I'm glad that you were able to watch the video that I made there. It was a little bit on the long side, I noticed. So hopefully I didn't bore you too much, but we really do a lot of great things in chemistry. Is everything that you've seen today, and we're all about hands-on, and it's my department and it's biology and IPH, and everybody here is really digging in with their students, getting some things done. The things that you watched in the chemistry video are things that you would do as a student in your day-to-day classes as you progress through the chemistry sequence. There's also an awful lot of opportunity in research. And so one of my research students, Christian Lund, is here, I believe, online. He's going to talk a little bit about the work done in my research lab. So I'll turn it away from me and let you take it away.
Yeah, hi, my name is Christian, and as Dr. Turk introduced me, I am one of his research students. I am a junior and double majoring in chemistry and neuroscience. So I know there was a lot to that video, but the instrument that I actually use is the plate reader. So this my research, unlike some of the others, was it's ongoing during the semester, so it's not a summer experience. Those are available. It's just for me, I do it during the semester. So what I did was I approached Dr. Turk and I just asked him about what he was doing. And then he talked to me about his research, and he already had a student working on the organic side of creating the molecules. So, oh, sorry, excuse me, I didn't explain what the research was. So the research is, there was also that freezer, if we saw with the H1N1 virus. So what we want to do is we want to create molecules that inhibit the replication of H1N1 in the body. So what he did and has been doing over the past few years is him and a bunch of other organic chemists have created a whole bunch of molecules, and we just have stockpiles of molecules. And my job is as the student is to then take those molecules, take the plate reader, and take the virus, and then see what happens. So what I do is I walk in. We have the we have a fluorescent substrate. The virus interacts with that substrate and fluoresces. So then we are able to determine the inhibition of the virus by the levels of fluorescence on the fluorimeter. So I have these plates that I come up with, and I put a whole bunch of that fluorescent substrate in there. I put the virus, and then I put the inhibitor. If everything goes well, what we'll see is that as the inhibitor concentration increases, the total fluorescence will also decrease. It takes a lot of trial and error, I'll say that. It's been a long road. This is my third semester. Unfortunately, I got cut short a little bit my first semester, but it's been a lot of fun. As you guys can probably tell, Dr. Turk has a lot of energy, so it's always fun to go in and meet with him, talk with him. He's been a really great resource. One of the things I really enjoy about doing my research is that when I went in, it was a lot of kind of hand holding. Like he would sit with me there, and we would look at the procedures and just kind of. It was a lot of him directing the process, but then over the course of working with him, it's become more independent, and I've gained the confidence, and I've gained the knowledge, and I've gained the skills to be able to lead the process, and then kind of gain my own specialty, and then what was I going to say here? Just be more contributing to the actual research, and I just feel like I am doing what is written on a procedure and just going step one, step two, step three, but actually understanding the process, being able to adapt the process, suggest things in the process.
That was great. Thank you very much. I appreciate all of our students and our faculty. So Dr. Karen Ball is here also. She's a faculty in our IPHS department. She didn't actually do any of the presentation, but I wanted to make sure you all knew she was here. If anybody has questions for Dr. Ball, for Dr. Harfman, Dr. Rowe, or any of our students, please at this time, feel free to ask them. We would love to answer any questions that you might have. You can just go ahead and unmute yourself and ask away. Does anybody have anything? Does any of our faculty or students want to add anything more before we head out if we don't have any questions from our students?
Is Dr. Turk still there?
I think he had to go back to class.
Okay. I just want to say that looking throughout the videos that your labs are just so amazing. And of the other colleges that I've visited to, it's nothing like your guys's. You guys are a lot more advanced and have a lot more student opportunities than the universities and colleges that I've already checked out.
Well, that's wonderful to hear. Thank you. We love to hear that.
Thanks, Kim. One of the things that I've always said, I remember several years ago, I was meeting with prospective students, and one of the parents, as we were going through the labs, one of the parents said, at the bigger universities, we see there are more pieces of equipment, right? And so, I'm kind of wondering, will my student have fewer opportunities to work with this equipment? And I've always said, no, absolutely not. Students at Alma will have more opportunities to work with that equipment, because it's as my colleague Dr. Harfman said in talking about the equipment in our labs, that is your equipment. We don't have graduate students that are here and postdocs who dominate the use of that equipment. That equipment is yours, and we view it as yours. So everything that you saw is there for our students to work with, and it will be there for you to work with should you join us.
Anybody else have anything, any questions or anything else to add? I do apologize. I know some of you had issues trying to join us. Just know that this has been all recorded, and so we will definitely share it back out with everybody. So if you were had to join us late, we can definitely get you the recording so you can see anything that you missed. But there's no other questions, then I just thank you all for joining us. I hope you got some great information out of this. We enjoy sharing it with you, and I hope you all have a great evening.